Hello everyone, we are going to study amyloidosis. What is amyloid? Amyloid is a pathological proteinaceous substance which is deposited in the extracellular space in various tissues and organs of the body in variety of clinical settings. So, amyloid is a proteinaceous substance which is deposited extracellularly. On light microscopy and with H&E stains, amyloid appear as an amorphous eosinophilic hyaline extracellular substance with progressive accumulation that encroaches on and produces the pressure atrophy of adjacent cells. To differentiate amyloid from other hyaline deposits, a special stain, Congo red stain, is used, which give pink or red color on light microscopy, and on polarizing microscopy green birefringent. So this is the microphotograph of liver, in which you are seeing pink red color deposition of amyloid in the sinusoids and in the blood vessels. This is Congo red stain, and on right hand side you are seeing the amyloid giving the green birefringent appearance under the polarizing microscopy. Now coming to properties of amyloid protein. Amyloid protein, first we will discuss physical nature of amyloid. Amyloid is seen under electron microscopy and it is made up of continuous non-branching fibrils with a diameter of 7.5 to 10 nanometer. It is important. It is made up of non-branching fibrils with diameter 7.5 to 10 nanometer. On X-ray crystallography and infrared spectroscopy, the amyloid has cross beta pleated sheet conformation. This cross beta pleated sheet conformation is seen regardless of the clinical setting or chemical composition and is responsible for the distinctive Congo red staining and birefringence of amyloid. So in figure A, we are seeing the amyloid fibers. An amyloid fiber schematically showing 4 fibrils, or it may have 6 fibrils in each fiber, wound around one another with regularly spaced binding of Congo red dye. Congo red staining with apple green birefringent under the polarizing light. Now the chemical nature of amyloid. Amyloid is 95% is composed of fibril proteins. So it is protein, and remaining 5% is comprised of P component and other glycoproteins. There are more than 20 biochemically distinct forms of amyloid proteins. Important ones are amyloid light chain, amyloid associated, and A-beta amyloid. The AL or amyloid light chain is derived from Ig light chains produced in plasma cells. Amyloid associated is derived from non-Ig protein which is synthesized by the liver. A-beta amyloid is produced from beta amyloid precursor protein and is found in cerebral lesions of Alzheimer's disease. So first we are going to discuss AL proteins. AL protein is made up of complete immunoglobulin light chains or amino terminal fragments of light chain or both. So basically AL protein is made up of immunoglobulin light chains. Most of the AL protein analyzed are composed of lambda light chains, but some cases may contain kappa chain also. The amyloid fibril protein of the AL type is produced from free Ig light chains secreted by a monoclonal population of plasma cells, and its deposition is associated with plasma cell disorders or plasma cell tumors. Now AA amyloid associated. Second major class of amyloid fibril protein. It does not have structural homology to immunoglobulins. It has a molecular weight of 8500 and consists of 76 amino acid residues. AA fibrils are derived by proteolysis from a larger precursor in the serum called serum amyloid associated protein. So, AA is derived from serum amyloid associated protein that is synthesized in the liver and circulates in high density lipoprotein association. Production of serum amyloid associated protein is increased in inflammatory states as part of acute phase response. Therefore, this form of amyloidosis, that is AA form of amyloidosis, is associated with chronic inflammation. So it is also known as secondary amyloidosis. So AL type in plasma cell disorders and AA type in chronic inflammatory condition. Now the A beta or amyloid protein. 4000 Dalton peptide. It is 4000 Dalton peptide that constitutes the core of cerebral plaque found in Alzheimer's disease and amyloid deposited in wall of cerebral blood vessels. So in brain there is amyloid protein deposition and the disease is Alzheimer's disease. A beta protein is derived by proteolysis from much larger transmembrane glycoprotein called amyloid precursor protein. So amyloid protein is derived from amyloid precursor protein and it is seen in Alzheimer's disease in form of cerebral plaques or deposition in the cerebral blood vessels. Next type is transthyretin. Transthyretin is normal serum protein that binds and transports the thyroxine and retinol. A mutant form of transthyretin and its fragments is deposited in group of genetically determined disorders referred as familial amyloid polyneuropathies. TTR is also deposited in the heart of aged individuals, that is senile systemic amyloidosis. So transthyretin is seen in familial amyloid polyneuropathies and senile systemic amyloidosis. Beta-2 microglobulin type, it is seen in long-term hemodialysis cases. In minority of cases of prion disease in CNS, the misfolded protein also aggregate in extracellular space. In addition, other minor components are also present in amyloid. The serum amyloid P protein, it may contribute to amyloid deposition by stabilizing the fibrils and decreasing their clearance. Coming to pathogenesis of amyloidosis. Amyloidosis, it results from abnormal folding of proteins which are deposited as fibrils in extracellular tissue and that disrupts the normal function. Misfolded proteins are often unstable and they self-associate, leading to formation of oligomers and fibrils that are deposited in the tissue. The diverse conditions are associated with amyloidosis may be that each results in excessive production of proteins that are prone to misfolding. The proteins that form amyloid fall into two major categories. First is normal proteins with normal proteins that have an inherent tendency to fold improperly. So they are produced in increased amount. When their number is increased, normal protein with tendency to fold improperly. And second is mutant protein, presence of mutant proteins that are prone to misfolding. So this diagram is showing pathogenesis of amyloidosis, showing the mechanism. So when there is production of normal amount of mutant proteins, example transthyretin, this will lead to aggregation of mutant fibrils or oligomers. And when there is production of abnormal amount of normal protein, like in plasma cell disorders in which monoclonal B lymphocyte proliferation is there, immunoglobulin light chain is formed, leading to AL protein formation. In chronic inflammations, there is activation of macrophages and release of cytokines interleukin 1 and interleukin 6, which will cause release of or formation of SAA protein in the liver, leading to AA protein formation. Coming to classification of amyloidosis. First is systemic or generalized amyloidosis, then hereditary amyloidosis, systemic senile amyloidosis, and localized amyloidosis. In generalized or systemic amyloidosis, first is immunocyte dyscrasias with amyloidosis or primary amyloidosis. Associated diseases are multiple myeloma and other monoclonal plasma cell proliferation. So major fibril protein is AL and related precursor proteins are immunoglobulin light chains, lambda type. Then in second is reactive systemic amyloidosis. Associated diseases are chronic inflammatory conditions and the protein is AA and precursor is SAA. Hemodialysis associated amyloidosis. Disease associated is chronic renal failure. Major protein is A beta 2M and the chemical related precursor is beta 2 microglobulin. Hereditary amyloidosis. The conditions are familial Mediterranean fever in which major fibril protein is AA and precursor is SAA. Second is familial amyloidotic neuropathies. Protein involved is transthyretin. Precursor is transthyretin and mutant ATTR is major fibril protein. Then systemic senile amyloidosis. In this, the precursor protein is transthyretin and the major fibril is ATTR. Then localized amyloidosis. In localized amyloidosis, there is senile cerebral amyloidosis, endocrine, medullary carcinoma of thyroid, islets of Langerhans is involved, isolated atrial amyloidosis. The associated diseases are Alzheimer's disease and type 2 diabetes. In senile cerebral amyloidosis, APP is the precursor protein and A beta is the major fibril. In endocrine disorders, calcitonin is the precursor protein and ACAL is the major fibril protein. In medullary carcinoma of thyroid, islet amyloid peptide is precursor and AIAPP is the major fibril protein. Islets of Langerhans and isolated atrial amyloidosis. The precursor proteins are atrial neuriuretic factor. Thank you.